the call.
Mm-hmm.
With this, I would like to welcome Professor Patrick Tan to the session. Very exciting to hear about the PRECISE initiative and all the work that you do. Before we start, I just wanted to share some notes on Professor Patrick, who is an executive director of PRECISE. He is currently overseeing the implementation of phase 2 of Singapore's precision medicine strategy, aiming to transform healthcare in Singapore and improve patient outcomes, and also gain some new insights into the Asian genome and data-driven healthcare solutions. I guess a huge accolade that during the COVID-19 pandemic, he was the program director of Operation Stronghold and established one of Singapore's largest COVID-19 testing facilities. And that it was together with the Agency for Science, Technology and Research, National University Health System, and Temasek Holdings. I guess more importantly, looking back at some of the accolades, there are many of accolades Professor Tan has. Started his undergraduate BA at Harvard and then moved to Stanford for completion of his PhD. And in 2018, he received the AACR Team Science Award as a team leader and the first team from Asia to gain this award. So many accolades and a lot of work that Professor Tan has done, but I'm going to stop now and introduce you. I'm very much looking forward to your talk.
Okay, thank you very much. I'd like to thank the Education Committee of the IHCC for inviting me. Let me share my screen and I hope that you can hear me. And you should see the full screen. Okay, let's get started. What I would like to do in the next 30 to 35 minutes is to present to you some of the approaches that we are trying to develop in Singapore to bring precision medicine from research into clinical implementation in a gradual manner. And I hope that you find it interesting, and I'll be happy to take any questions after the presentation. So let's move on to the next slide. I don't know how many of you have actually been to Singapore, but I wanted to give a few snapshots of the country, because I think that for many different of these population-based programs, one really thinks about what are the unique features that one's population or one's country or one's geography can bring to bear that ultimately will be part of a global community like the IHCC. So here we are in Singapore. It's a country in Southeast Asia. It's actually a very small country. It takes about half an hour to get from one point of the island to the other point of the island, and that's driving the long way. And in general, if you look at the left-hand side, we have a population size of between 5 to 6 million individuals, of which the majority are what we call residents, meaning they are long-term residents in Singapore, and they stay for many, many years, sometimes decades. On the right-hand side is Singapore's ethnic composition or ancestral composition. We have three major ethnic groups in Singapore: the Chinese, the Malays that represent Southeast Asian, and as well as the Indian populations, as well as a number of other races and ancestries. This has remained fairly stable. So in general, I think what is quite interesting for Singapore is that it's one of the few countries where one can then begin to compare genetics, lifestyle, between different particular ethnic groups and really look at how these impact on healthcare outcomes. And there aren't that many countries in the world, particularly for this particular composition, where you can do it all in a small, restricted geographical area. In terms of the overall age of the population, I think we are facing a situation where the overall population is aging And that is an important factor because that's actually driving some of the pressures in our healthcare system that I will describe in the next slide. So Singapore actually has done pretty well in terms of our healthcare outcomes. We spend about 4 to 5% of our gross domestic product on healthcare. And we have one of the longest lived populations in the world, around between 80 to 82 years old. But there are many pressures that we are facing because of population aging, more chronic disease, rising cost of therapies, that actually we are beginning to see more and more demand for healthcare services, particularly at late stages. This refers us to services in the tertiary care hospital. And clearly, the COVID pandemic did not help that. Many people did not get out and exercise. So many people delayed seeing their doctors in the clinic for managing some of their conditions. So this is actually beginning to, the population actually has gotten worse in health over the COVID pandemic. Ultimately, this has puts a lot of pressure on our healthcare spending, and there's a significant interest in the government on how we can continue to deliver good healthcare outcomes, but mitigate some of the spending rise, or else this will cannibalize from other areas of social need, education, research, social infrastructure, and so on. So this is the situation that the Ministry of Health in Singapore is facing, that we are seeing rising incidences of chronic disease, usually manifesting in our healthcare sector as by late stage. And this is resulting in increasing healthcare costs. And so this is driving the Ministry of Health to try to think about new approaches to deliver healthcare. And one of these that I'll talk about today is the role of precision health or precision medicine, where we're trying to use information and data about populations at different points along an individual's health and disease trajectory to be able to intervene. If you're healthy, can we predict which risks, disease risk you are going to be susceptible to, and can we intervene, prolong health span? If you have a disease, what is the disease subtype that you have that's molecularly defined, and therefore we can then begin to ascribe the right therapy for the right condition. And so I'm sure that all of you have seen different aspects of this slide, but in general, what we're trying to do is to have a national framework where we can collect data relevant to an individual's or a group's health outcomes, family histories, which essentially is their genes that they're born with, that they transmit, their lifestyle factors, medical histories and access to medical care. And the program that I represent, called Precise, has a mandate to collect data that's relevant to the population of Singapore, which also is relevant to a significant proportion of the larger Asian diaspora. One of the reasons why we're doing this for Singapore is that, as I'm sure that most of you know, information about genetics, lifestyle, disease conditions from populations outside the regular Caucasian Western populations is still underrepresented. So this is represents a national healthcare need that we want to be able to address, but also by doing so, can we also begin to contribute towards the larger global landscape in this space? So the program that we've developed is called the Singapore National Precision Medicine Program, or NPM. Is a 10-year program divided into 3 phases: phase 1, phase 2, and phase 3. We have finished phase 1. We're in the middle of phase 2. And what I'd like to do in the next part of the talk is to tell you a bit about the approaches that we took, some of the findings, some of the aspirations that we have as we go from phase 1, which is 10,000 individuals, phase 2, 100,000 individuals, and then phase 3 that we're going to start the planning process for probably at the end of the year, where we're going to be looking at phenotyping about at least 1 million individuals. So let me start off in phase 1 and what did we do for them. So when we started the program, we wanted to design milestones that could be generated as a resource that many people would find useful, but which would represent stretch targets that would be something that we could do with challenges, but it was doable. And so what we decided to do in phase one was to establish a reference genomic database of Asian healthy normality. And this underpins the basis by which one can then compare disease patients to identify disease traits. So we call this phase one database SG10K Health for 10,000 individuals, and it comprises healthy individuals upon recruitment. They are actually assembled from 6 existing research cohorts, mostly in the ranges 18 to 84, but we also have a very interesting newborn cohort where these samples were sampled at the point of birth. We have a mixture of different ethnic groups, Chinese, Indians, and Malays. And what we did was to collaborate with local sequencing vendors to sequence a whole genome sequencing of these 10,000. And in addition, we also we put them all on DNA methylation arrays. So we have epigenetic signals. We're still analyzing those, as well as a limited number of phenotypes, because the basis of phase one was really about having this reference genome of Asian genomic normality. So we got that done. And in addition to the sequencing and the science, there were a lot of other aspects that we needed to put in place to prepare the Singapore ecosystem for the eventual rollout of precision medicine. I'll highlight a few of them in this slide. So on the top left was to pass standard codes of practice on when genetic tests can be delivered and when do you need things, for instance, like pre-test genetic counseling, post-test genetic counseling. And we were also able to work with the Ministry of Health and the life insurance companies to put a moratorium on the use of genetics for insurance underwriting. We felt that this was very important. The US has it, many countries in Europe has it, not so many countries in Asia. And so this is important so that when they see their genetics, when they see that they have particular mutations in the genome, they can act on it without being fear of having discrimination. Other aspects that I'll get into was a robust public engagement effort where we were engaging the public on the parameters by which we could actually begin to collect data about them and the attitudes that they would have about sharing that sort of information from industry, from academia, from local institutions as well as overseas institutions. And I have some interesting points about that at the end of the talk. And some of the other aspects I'll get into when we in the subsequent slides. So one of the things that was very important to us was to have a basis by which this data that was generated would not remain siloed, locked in a secure server, one room vault somewhere, but actually accessible to the community. And so we invested quite a bit of resources into enabling the information to be accessible by the community. I highlighted 2 of them here. On the top, there is a web service that allows you to access aggregated information. This is available to anybody on this call. All we need for you is to be associated with an institution. You can put in an access request and you can actually access it, no strings attached. And this allows you to browse variant allele frequencies. Those of you who know how to use the Nomad browser will find this very useful. But we also have other features like an imputation server. Snipshot 3D is quite interesting, where we've been able to map all of the different variants to 3D crystal structures, as some of you may know, generated from programs like AlphaFold. And so I think this allows people to use these sorts of tools. But we also have data access application processes by which one can then put in an access to access the individual level data, and that also gets triaged by a data access committee. And this is open to both academia as well as industry. So I think, and here are some examples of applications that we have approved. So our main purpose here is to have the data, but we want to get it out there in order for people to make use of it, to make discoveries and to deliver insights. So what can you do when you sequence 10,000 Asians? So the first thing is that we can begin to look at genetic diversity, and I'll highlight this. This is a paper that was published in Nature Genetics just a couple of weeks ago that describes the program and some of the larger high-scale details of SG10K health. So we see interesting patterns like this. If you look on the left-hand side, what we've done here is to map individuals from the SG10K health database from Singapore onto the population structure of the world using the 1000 Genomes Project as a baseline reference. And if you just follow my pointer, if you look on the right-hand side, the Singapore Chinese in red overlaps quite well with the 1000 Genomes East Asian cohort. So this shows that it's quite representative. On this side over here, the Singapore Indian population maps quite well with the 1000 Genomes South Asian population. So we have representation of the Chinese population, of the Indian population. But very interestingly, in green are the Singaporean Malay genomes that actually show very little overlap with other currently represented genomic populations in the world, suggesting that this is a really novel population that is still very understudied. And because of a lot of these new genomic variants, we see millions of new genomic variants not currently represented in standard genomic databases. So there's a lot of avenue for further discovery here. In a companion paper was published, one of the group looking at medically actionable variants looked at the slice of the genomes that comprises actionable medical genes. And here some of the data where you can see that there are in red are the Chinese genomes, in blue are the Indian, and in yellow are the Malays. And there are certain genes where the risk variants are there and they are equally distributed among the three different ethnic groups like BRCA2. But there are other ones like LDLR, which obviously some of you will know that is causing familial hypercholesterolemia that's much more prevalent in the Chinese compared to the other two ethnic groups. And we can see medically relevant genes from a variety of different conditions, including cancers, cardiovascular disease, neurologic disease, and others. So one of the interesting aspects that we want to be able to do is to think about how can we intervene for these individuals that have very strong germline actionable genes. And a part of the program is to be able to use aspects like sequencing to be able to identify these groups that are high genetic risk of disease. We need to be able to pass legislation and the right processes to protect these individuals, to reduce the inequities they may face arising through no fault of their own, because nobody asked to be born with these particular gene variants. And finally, we want to be able to think about the right interventions and treatments, some of which are actually inexpensive, but you must start them early to be able to uplift these groups so that we can give them as equal shot at success and realizing their human potential as the rest of the population. So that's one of the directions that we think about when we undertake this particular program. Obviously, when we want to deliver these results, we had to develop a clinical workflow where these initial results that were sequenced in a research-grade setting could then be flipped into a clinically accredited, validated setting with the appropriate genetic counseling and delivery of results. And we were able to do that. This was done in phase 1. We returned to about 40 to 50 people. This will obviously scale up when we do phase 2. One of the interesting things that we see is that because many of the genomes that we sequence are underrepresented, we begin to see that this challenge of variants of unknown significance is quite striking, and that's even for variants that are found in protein coding genes. So just to give you an idea, that for every one pathogenic variant that we can classify using ACMG guidelines or bioinformatic algorithms, we actually see 4 variants that we are actually not quite sure if it's benign or if it's pathogenic. And just to give you some of the numbers, there are a number of variants that the group that I just did the study looked at, that hundreds of variants that just fail the criteria to be defined as pathogenic. That means that you're probably underreporting the individuals that actually are going to have a genetic disease related to these particular conditions. And so one of the things we think about are what are the sorts of new approaches that we can do to be able to functionally validate these variants at a level where at some point the bioinformatics actually runs out in terms of its utility. And I think this is going to be one of the new challenges. There's a problem, but the opportunities for some of the newer cellular high throughput technologies that can then begin to do functional analysis of these variants at scale. So I'll just give you one example of what we are exploring in a limited way on how we're doing this. So this is an example of an individual that had a VUS, a variant of unknown significance, in a gene called MYH7. And in that individual, what we were able to do was to work with the National Heart Center in one of our leading hospitals to be able to generate iPS cells from that individual. And once you have those iPS cells, you can then begin to differentiate those cells into different tissue types like cardiomyocytes. And then you can do functional testing to ask the question, for individuals that have that variant, do they show a cardiovascular phenotype? And more importantly, using CRISPR, you can then begin to revert that variant back to wild type. And the expectation then is that you should not see that abnormality. So it's kind of working. Just to give you one data point here, that compared to wild type cells, just look on the right hand side here. If you take the iPS cells converted to cardiomyocytes and you look at relaxation times, you actually see that there is a defect in the relaxation times, but that goes away when you take the same cells and you CRISPR correct them into the normal variant. Now, obviously, this cannot scale to hundreds of thousands of individuals, but we think that this is an opportunity to create, in a very fixed way, particular banks that can then be used for subsequent functional type testing. And this is where the importance of having the ability to recall individuals for further testing and further phenotyping becomes very important in many of these cohort studies. Another interesting thing, just as a interesting story, is that because it's whole genome sequencing, you can actually begin to look at other interesting questions. So this is a paper that's coming out in Nature Microbiology, where the group looked at the question of, for these sequences that are all from blood, do they see traces of a blood microbiome? Meaning that is there a component of microbes that's coexisting in the blood? And the reason why this is a topic, it's a very topical question, because traditionally blood has been considered as a sterile site, but a number of papers have come out suggesting that in both healthy as well as in these conditions, there's a microbiome associated with these particular individuals. However, we looked at it, and what this group said was that after going through stringent bioinformatic decontamination processes, they actually see no species, and many of the species that you see are very transient, suggesting that they're not stably associated with the blood niche. And so we do not see any evidence for a common blood microbiome based on our Singapore data. So I think this gives you a sense that when you do the sequencing, you can actually look at a lot of many of different aspects. So again, this is why we want to be able to get the data out there for as many people to look at as possible. I'm now going to go on to what we're trying to do in phase 2, which is, as I said, ongoing. Because phase 2 is 10 times the size of phase 1, we needed a central entity to coordinate this across the different entities involved in the program, the hospitals, the medical schools, the government agencies. And therefore, we established PRECISE, or Precision Health Research Singapore, as a central entity to coordinate this entire program. The goal of the phase 2 is to really think about how we can converge cutting-edge research, and I'll tell you about this precise SG100K cohort that we're setting up, along with clinical implementation, where then we can begin to think about findings that emerge from the research and how can we bring them into and mainstream them into clinical practice and clinical implementation, which is actually carries a different set of challenges compared to research. And I'll try to describe them as we go into this phase. So the cohort that we're trying to establish is a prospective longitudinal cohort because if you take what I said before, we're interested in disease prevention. And so having a longitudinal cohort that we can monitor and track for outcomes is going to be very important. It's 100,000 individuals consented, and each individual goes through a 6-hour phenotyping exercise that is essentially the equivalent of the UK Biobank that you can see on this slide over here. And we've added in a few other phenotypic measurements. All of these individuals are currently undergoing whole genome sequencing, and I have 1 or 2 slides on that. And if you think about it, so the baseline phenotypes when they're healthy, you can collect them, you have the sequencing, but ultimately these individuals then will progress to develop disease. And that's where the usage of accessing these individuals through the electronic health records nationwide allows us to pull in clinical outcome data regardless of where these individuals encounter the Singapore healthcare system. And it's not just the clinical data. The Health Promotion Board, I have 1 slide on that, has lifestyle data. So they are running programs on wearables and Fitbit monitoring. And that information can also be pulled into this particular database. So the whole idea is to have a long-term national resource that can power current and future clinical and biomedical research. Because in the interest of time, I won't descend it, but essentially precise SG100K is a combination of 2 cohorts. 1 cohort is retrospective, 50,000 people collected already, 15-year follow-up. On the right-hand side, another 50,000 that's prospective that has extended phenotyping. The sequencing is done at scale in a public-private partnership with Illumina. We've established centralized labs for DNA extraction, public awareness, and a dashboard here to monitor sequencing as it goes through. Here are some of the metrics. Our average coverage is about 41x, and this is run through the Dragon pipeline that outputs 9 different classes of variants. So this is a tremendous amount of information that we can use to do genotype-phenotype associations. What this slide shows you is that when you take the information on genetics and link them to clinical data from the Ministry of Health. So this is data that's not collected as part of research, but data that's collected as part of an individual's routine health care. You begin to see the expected associations. So what we've done here is to stratify the 10,000 individuals by a polygenic risk score for different traits, and we've associated it with the blood biochemistry values done as part of routine clinical testing. And you can see that there are very, very strong associations. The p-values here are in the order of -50, -97, and so on. So we see this, and as we begin to enlarge this, we'll be able to access and to have many different clinical phenotypes as we bring those in. I want to move on to the other part, which is, if anything, even more important than the research. It's how you take testing like genetic sequencing, could be whole genome, could be targeted panel. How do you bring them into the healthcare sector in a fashion that ultimately makes sense for the country? And so what we're doing here is that we're running clinical implementation pilots where you can then, these are very focused clinical scenarios, and we have 5 of them. Familial hypercholesterolemia, polygenic risk scores for breast cancer, inherited cancers, and so on. And trying to work with clinicians and healthcare systems to develop clinical pathways where these people can be tested, and based on the test, different actions are done so that one can then create health economic models that can ultimately ask the question, did this provide value-based healthcare for the country? So what we have is kind of a house. We have the clinical implementation pilots in the middle, but what we are doing to support these are platforms that will drive down cost. So common platforms for clinical grade sequencing, genotyping, so that all of these will go through one platform so that we can begin to drive down cost, which is very important for health economics. And secondly, a confederate of health economic individuals that can look at the data, develop the models, and ultimately take that to the cusp of clinical implementation for approval by the Ministry of Health. I won't go into that. So let me just last few slides before I close my talk today. The other aspect that is important in the program is how can this sort of public investment catalyze industry interest? Because I think that ultimately to be sustainable, there has to be an interaction with industry. And so here are some interesting public engagement efforts in terms of the willingness of the Singapore public to share their data with different organizations. As the organizations that are further to the right are the ones that have the most trust. So we have government agencies, universities, pharmaceutical and biotech companies. Is a bit of a drop, but also it's not too bad. But interestingly, private insurers actually are quite bad. So obviously, this tells us a lot in the sort of the assurances that we need to build in order to make sure that we don't get a backlash from the public. And overseas universities is quite interesting, but I think that we need to do a deeper dive on what exactly are the concerns there. But all of this individual ultimately tells us a bit about when we want to work with a company, what are the sorts of parameters that that the company needs to fulfill in order to be able to access some of the information about the Singapore population. I won't go into this because it's a conjoined graph, but very fascinating. I can talk about this later. So one example of I think a very fruitful collaboration with industry is the public-private partnership with Illumina. I talked a bit about the sequencing, but in addition to that, some of the aspects that they're bringing to the platform are their new cloud platforms for enterprise-grade genomic high security analytics, bringing in companies that can use their software toolkits to develop genomic applications, and also enlarging the teams in Singapore looking at artificial intelligence and machine learning for precision medicine data that ultimately will increase the teams and the expertise in Singapore for this particular area. This is obviously coupled to a workforce development where they're funding PhD positions, graduate student internships for people to understand how precision medicine research can be done. This is one example. We have others of the sorts of things that we can establish through these sorts of national scale programs. So this is my last slide. Just as some of the competitive advantages or the value proposition that Singapore has that I won't go into. And so I think that we are currently in the middle of phase 2. We seem to be doing a reasonable job. And I would like to thank everybody here for coming to the talk, and happy to take any questions. So thank you very much for your time. Thank you.
Thank you very much, Professor Tan. That was really insightful, and a lot of work. And having been involved with a trial or a cohort of 202,000 women who have been linked to various registries and had the longitudinal data and sample, this is quite a major effort. So I guess the first question that I had was about some of the data that comes through here, and you've used the example of MIH7 VUS in cardiomyopathy, and also the potential to do CRISPR editing. So as most of those familial hypertrophies would occur, I guess would first present with sudden death in quite a young individual, how do you foresee using the data to screen the population, because you have, I guess, a smaller population than the UK? Are there any ways that you have already thought about how that could be implemented in Qatar, whether it's in the neonates, and just be quite interested to hear.
Yeah, I think that the value of the genetics is that you can detect these people that are at risk before the symptoms develop. So for instance, familial hypercholesterolemia, if you want to intercept the disease trajectory, you really must start when the patient's at a very young age, not when they're 40s, when there's already been a lot of high cholesterol, right? So what we're trying to do is to explore working in the Ministry of Health to be able to screen at a much earlier age. So in Singapore, when one goes for the standard testing for high blood cholesterol, it's usually on the age of like 30 to 40. So for these individuals, that's way too late. And so we want to argue that no, what you're going to do is that you want to be able to screen these individuals. But obviously, when you sequence, you will not just sequence for one gene, you will sequence for a whole host of different types of patterns. And so we can detect those. And for those that we need to be able to intervene. So as I said, some of these treatments to improve health are not super expensive and not super high tech. Some of them can be supplements. So I think that's one point. The second point is that we're not advocating that everybody has iPS cells done. I mean, I think that's a bit too much. But I do think that what a lot of these programs will uncover is a need for functionalization on millions of variants. And that's not just in the coding. Once you get into the non-coding space. And so I think that there will be a whole new science on how we can validate and test all of these particular variants. Some of them must be tested in the tissue of origin, right? That if you look at expression QTL, so we'll need to, and so iPS cells are attractive because then you can differentiate them into different tissue types that are of interest to your particular disease. So I think that's it's sort of like the next wave of technology development that has to happen for this sort of population cohort studies to really go to the next level.
Thank you. Opening the floor to questions or comments from anybody on the call. I've been looking at the chat, but I don't think we have anything.
Hi, Patrick. Can I ask a quick question?
Yes, of course.
Hi. So you mentioned early on in phase 3 of your NPM program that one of the things you're interested in doing is creating made in Singapore tools. And I believe your database was one example of that, but are there other examples of the tools that you will be making during this process of NPM? And will you be exporting those tools to the rest of the world?
Yeah, I think that's the vision, right? I remember vision, like the reality, maybe, but certainly, as I alluded to, and it's to a first approximation that the population in Singapore the findings could have applicability to much larger populations across Asia, right? And so there is this opportunity, right? So if we develop, for instance, a diagnostic, so based a carrier panel based upon Singapore genomes that has content that's not reflected on current Western panels, that could be a product. And then that could be exported to other countries. And now for that, we need interactions with the private industry. Because as the Ministry of Health in Singapore, I can't go to other countries. And so I think that there is this opportunity to be able to think about how we can develop a economic ecosystem where these individuals can then, and and to be honest, once you export it, the market sizes are much larger than Singapore, right? And then and then this could then be used in a way to make the program sustainable in the long run, because some of the a revenue could then be brought back. So I think those are some of the aspirations, because you can't rely on public funding forever.
Thank you.
Can I just go back to the longitudinal sampling and data that you have? How often will you be, or are you collecting this? Is it every year or?
Yeah,
every
so we have the ability to recall individuals for longitudinal follow-up. We talked about, but it's quite expensive, and you always get a dropout rate and so on. So what we are investing in currently is that, so long as we know, and this is all under full informed consent, so long as we know the identity of the person, so long as they develop any disease or any symptom and shows up in any hospital in Singapore and has a medical record, we will be able to extract that information. So if a person comes in and has a cholesterol test, or has X-ray, or has an ICD-10 code, we will be able to. So in a sense, the longitudinal follow-up is done in the background, because as these individuals develop disease and symptoms, it doesn't preclude us from investing in a smaller set for very targeted types of recall types studies. But I think on scale, the appeal of this is that many of the healthcare incidences if they occur in the Singapore healthcare system, we will be able to monitor that, and that's the basis of the longitudinal follow-up.
Right, so it's actually the linking of the cohort, and there'll be longitudinal follow-up, but not samples that are taken at a particular interval and predate diagnosis. Diagnosis of cancer, CVD, right, or otherwise.
Right. We collect all samples at baseline. So we collect blood, we collect stool, we collect microbiome. So that's one time point. Right. We have toyed with the idea of repeat sampling, but we have not had the courage to actually do that yet.
Great, thank you. I can see that Roger Glass has a question. Please go ahead.
Yeah, Patrick, that was a fabulous presentation. And I had really two questions. One is, do you have any pushback on people having their genomes sequenced or in a bank, or privacy, those kinds of issues? And second, we at NIH have been working a lot in Africa, where it's a push. People don't appreciate what's in their genome and how that can be practical. What have been the most practical, cost-effective diagnoses, interventions that you've been able to make that have been able to allow you to convince policymakers that this is a worthwhile endeavor?
Yeah. So I would say that there's constant public engagement. We have focus groups. We have consultations on this matter that can probe overall attitudes. At the end of the day, it's a question of, does the public have trust in the institution that's the custodian of that particular data, that they will use it responsibly? And as I showed you, one of the, rightfully or wrongly, the Singapore government has great faith in the Ministry of Health. So I think that, so long as it's under that there is it's going to be used responsibly for the betterment of the Singapore community. I think there is an overall sense that that's okay. That is an important relationship that needs to be protected, and so we invest a lot in making sure that happens. And personal stories of individuals that were diagnosed early, or an individual with a cancer mutation. These can be very, very powerful types of stories and messages. And once in a while, our Minister of Health has come out to actually talk about the importance of this. So, as I said, I think those are the sorts of things that have reassured the public that it's not that this is actually done in a responsible, considered way. But it's not it's something that we don't take for granted, and we think that this is it's cardinal to the whole program.
Mm-hmm. Right, in the earliest diagnoses that have been functional, I'm thinking about Africa. Well, what would be the first diagnoses that would impact care and care and prevention?
So, I mean, I have less familiarity with Africa. One of the things that actually we were able to show, it's a bit circuitous, but actually, the fact that we had a reference database of normal genomes, this not counting the actionable mutations, right, allowed us to show that if you have a disease patient with a genetic disease, by comparing it to the reference genome, you could improve your clinical efficiency by 40%. Because you remove all of the variants of unknown significance that are just there because of the fact that you don't have those particular databases. And so there was a lot of enthusiasm by the clinicians to use this sort of information because it just made their life easier. They didn't have to filter through so many variants to look at. So it was really positioned as this was the sort of research that's going to help a lot of different types of conditions. So that was one thing that was quite compelling. And I guess at the end of the day, it's a question of does the clinical community embrace this sort of work? And then obviously we had other things like the diagnoses. So we had BRCA, we had familial hypercholesterolemia, and so on. And those things also. So I think that and that works more on the public side, right? But at the end of the day, you want to also be able to convince the clinical community that this is worthwhile doing.
Good, thank you.
Thank you.
Don't know whether anybody else has any questions, but I just wanted to ask, you mentioned internships, and I guess we're looking at how we can, I guess, cross-pollinate between the cohorts. And I don't know whether there was an opportunity for somebody from another cohort to perhaps, you have wealth of data, I guess, to be analyzed for quite some time. Data will be accumulating. Are there any opportunities that we could be discussing for those quite early in their career, either PhD students and postdocs, to come and work with you? Is there something that's...
No, definitely. No, Singapore is a small country. We do pride ourselves on having open borders, right? And I think there are many opportunities. We actually there are actually, depending upon the institution, we actually offer certain schemes for funding students, postdocs, and so on. Under different types of frameworks where people can come and spend some time in Singapore, learn, and then at the right time either stay or they can go back to their home country. So I think that that is something that we do have in place. And yeah, Singapore's a nice place to live. So for a few years.
Okay, great. Well, hopefully you don't mind if we get in touch and just explore some of those opportunities. But I think on that note, I would like to thank you on behalf of the webinar series and the committee on an excellent talk. Before we go, I'm just going to hand over to Latifa to announce the next webinar, and thanks to everyone for attending.
Yeah, thank you so much, Alex, for moderating this session, and Professor Chen for this great presentation, and attendees for your participation. Yeah, I just want to mention that we are going to have IHCC educational webinar series, Qatar Biobank Showcase, that is scheduled for March 1st until 9. And this is the first time we are going to do a series of webinars. So I put here on the chats the link to register for this webinar series. And also, if you need more information on this biobank webinar, this exciting event for us and for you, please check the description that is under the link that I put on the chat. Thank you very much. And please, if you have any question, don't hesitate to contact us. I can put also my email contacts here.
Nice job organizing, Latifa, as always.
You're welcome.
Lastly, Latifa, I'd add for people that want to find out about upcoming webinars, upcoming information on other activities that the International 100K Cohort Consortium is doing. Our website at ihccglobal.org is updated regularly by you and others. So you can always go to the website and find out the latest goings on and news and updates on everything that you hear. If you like the webinars, we have them at least every month, sometimes even more than once a month as we've been doing lately. So please join. We love your participation.
Yeah, thank you, Scott. As a matter of fact, in addition to this biobank showcase that will be presented by Qatar Biobank team, so we are also having another webinar by the end of March, and also others, as you said, we may have like 2 webinars per month on the next months for this 2023. So exciting events are coming, so we're looking forward to your participation. And I put also my email in case you have any question. Please don't hesitate to contact me directly. Thank you.
Thanks, everyone. Thanks for joining.
Thank you, everyone. Thank you, and have a nice afternoon, evening. And we'll see you in the upcoming series, IHCC Educational Webinar Series. Bye-bye.
Thanks, everyone.
Thank you.